You feeling beat up, or are you just feeling like overworked?
Really beat up.
Okay.
Lots of injuries happening this year.
I feel like you guys have, whenever I'm in like the training room, there's always a whole bunch of football players.
Have I shown you anything wrong?
No, just doing that.
All right. It's 12:30 by my watch.
Fast on it.
Yes.
Yeah, I know, Jamie. Sorry. You're gonna survive today, though.
Okay.
All right.
Alright, now we're going to
You're not gonna light my brain on fire?
What?
You're not gonna light my
When you
brain on
get
fire?
it.
No.
I'm
No, I'm not gonna kill them and throw them out the window.
parents right now.
All right. Today, the only people I want to hear from are the high toppers, which none of them are here. The athletes and half-leets, and athletes, high toppers, and beauty and the biologists.
None of the high toppers are here.
I know, so it's up to the rest of you.
Just say you want everyone but me to talk, just say it.
No, I also don't want to hear from these guys.
Just say it.
I don't want to hear from you guys.
Why not?
Because you're way ahead on points. We need to give the other people
Tell
a
the
chance to
truth,
catch
wait,
up.
every time we speak, we do talk, there's no point in it.
Yeah.
Here
it's
he is,
terrible.
here he is. Yeah.
I came in before class and I put points on people's things. You'll see that the point totals have changed based on the last couple of
We
days.
were chatty Cathy's the last week.
What's that?
We were chatty Cathy's, or.
You have been,
chatty
which is
Cathy's.
why I redistributed some points.
What's that on your arm?
Really does talk about
Like a tattoo, not on top.
You do talk about
Exactly.
I mean,
I personally
I think that's what-
It looks scientific.
All right, focus, focus, focus. We're talking about nitrogen cycling. Nitrogen is a complicated nutrient cycle because it has a lot of different kinds of transformations. So before we move forward, what difficulties are you having with understanding how the nitrogen cycle works? Sonia, do you have a question?
Are we gonna talk about VNet all before the paper is due? Because I have like a question.
We have been talking about Veen et al. This
Okay.
entire 3 weeks.
Okay, okay.
No,
Why-
as soon as we're done with the nitrogen cycling, Dr. Judd is going to talk to you for a little while about human impacts, mainly eutrophication due to nitrogen cycling, and then also prairie decline, and then the rest of the class period is going to be on talking about any questions about the test and any questions you have about Veen et al.
Alright.
But we have been talking about Veen et al. For the last 3 weeks.
Well, I didn't know if I was allowed to ask a VNet all question or not.
Yeah, sure. Can you save it until
Yeah.
towards the end?
okay.
Okay, all right. Questions about the nitrogen cycle? We will get to it. All right. So we were talking about the rhizosphere and how mycorrhizal fungi extend the reach of the plants out into the soil by extending their fungal hyphae, their fungal mycelia, and I think I used hyphae incorrectly. They're fungal mycelia, these long extensions. And that gives the plants more access to the volume of soil that they can access to bring in nutrients and water and things like this. So that's one of these useful associations that plants have with other organisms. The transformations that we've been talking about so far with reference to nitrogen have been inputs and then intrasystem cycling. We haven't talked about the ways in which nitrogen leaves ecosystems. And so the last couple of things we need to talk about are the exit of nitrogen from terrestrial ecosystems. The first of these is denitrification. We start with nitrate, which is this super useful form of nitrogen that plants can absorb really easily. And we basically convert that into atmospheric nitrogen. Any guesses as to what kinds of organisms do this down in soil? The organisms we've been talking about so much in the nitrogen cycle.
bacteria.
Bacteria, not fungi in this case, but bacteria. Good. Yeah, so this is an anaerobic process. It doesn't require oxygen in order to proceed. This is largely done by bacteria. The rate depends upon the bacteria being in an anaerobic environment. It also depends upon, obviously, the supply of nitrate, and then also the amount of carbon, because this is a carbon-intensive transformation well. So this is how nitrogen gas gets back into the atmosphere, is through the activity of these soil bacteria. So hopefully one of the things that you've noticed as we've been going through this is that bacteria are hugely important in the nitrogen cycle. If we didn't have bacteria in soil, we would have real difficulties in making nitrogen available to other organisms, because they've been every step of the way. Volatilization is just transfer of nitrogen back to the atmosphere in the form of burning up biomass. And so this becomes very important at Kanza, because whenever they do a controlled burn at Kanza, a lot of the nitrogen in the biomass doesn't actually go into the soil, it actually just volatilizes into the atmosphere. And so we basically take ammonium and convert it into ammonia, and that ammonia is gas in the atmosphere. Once again, it depends upon aspects of the supply of the reactants, but also depends on things like pH and things like this as well. With fire, it doesn't take a particularly hot fire to do this volatilization. This is a fairly low temperature sort of reaction, and so this is why you tend to lose nitrogen in systems after fires. And then the last output is leaching. We've talked about leaching before. Somebody from beauty and the biologist give me a definition for what leaching is.
It's when like water takes nutrients out of the soil, like nutrients that plants need to grow.
Okay.
Because it has a chemical bond to it, right?
Yep, it does, because water is a polar molecule, so it will latch onto positive and negatively charged ions and drag them through the soil. This happens a lot in disturbed systems where plant uptake is reduced. So if you have a lot of water flowing through the soil because the plants aren't picking that up and blowing it back into the atmosphere through transpiration, all that water flowing through the soil just leaches the substances out. You also have large losses in nitrogen where there's an excess of nitrogen. And so this happens a lot in agricultural systems. So when Dr. Judd talks about eutrophication, a big part of eutrophication is actually just nitrogen being lost from agricultural systems where farmers apply more nitrogen than is actually needed onto crops, and that excess nitrogen leaches out. While negative and positive ions are leaching out, ions of the opposite charge can also be leached out as the ion that we're interested in exits as well. So both positive ions and negative ions both can leach out, and the more leaching you have, the more leaching tends to occur of other things that you might not actually be tracking. So we can make a model of the nitrogen cycle at Kanza if we wanted to. We would have a bunch of pools and a bunch of fluxes, but we wouldn't arrange it this way. We would arrange it more like this, where we have soil. And you can see that I had to make my soil box much bigger in this representation, not because there's a ton of nitrogen in soil, but because there's just tons of things going on in soil. And so what this figure encapsulates is all of these pools and all of these fluxes. And so this is one of the reasons that the nitrogen cycle is so challenging, is that there's just a lot to keep track of. So how do we simplify this? Well, we can think about inputs into terrestrial ecosystems. We can think about intra-system cycling within the system, and then we can think about outputs. So let's start with outputs, because we just talked about the outputs. What are some outputs? Athletes.
Like what we just talked about?
Yeah, what we just talked about. Okay, volatilization, burning up and going into the atmosphere. What else?
Gentrification.
What?
How do you pronounce that?
Denitrification. I thought you said gentrification.
No, you know.
Denitrification.
I'm illiterate, isn't it?
No, that's fine. What else? there were three outputs that we talked about. The last one, what was the last one? Has to do with water. Leaching, good. Okay. What is up there in terms of inputs into the terrestrial ecosystem? Somebody else?
Death,
Hmm?
death.
That's more in intrasystem cycling, and I'll explain why in a minute. But it's already there because the nitrogen is in the plant where the animal then dies. So the question is, how does it get from wherever it is, where it's not in the terrestrial ecosystem, into the terrestrial ecosystem? Sonia is raising her hand. David? Input.
What about atmospheric deposition? Is that like?
Atmospheric deposition would be one, yep, sure.
Yeah.
Good. What else? Where is the inaccessible nitrogen found? Gigantic pool of nitrogen that is largely inaccessible to organisms. Not the ground. I mean, there's a lot there, but it's super accessible. Yeah, mister, who was that? Oh, Sonia.
Sorry.
Don't apologize. So where are we going to get, how do we get that nitrogen out of the atmosphere into organisms?
Nitrogen fixation.
What's that?
Nitrogen fixation.
Nitrogen fixation.
Yeah. I said peanuts, yeah.
Peanuts? Oh, yeah.
Soybeans.
Yeah. Soybeans, peanuts, soybeans, all those things.
Peanuts.
So there are relatively few ways of getting nitrogen into terrestrial ecosystems. Nitrogen fixation, and then actually this is not a flux that is up there, which is this deposition. So all of these other fluxes are basically then what? They're not inputs and they're not outputs, so they are intrasystem cycling. So ammonification, nitrification, assimilation, denitrification. Oh, not denitrification. Denitrification is an output. Ammonification, nitrification, assimilation, excretion, litter fold, death, decomp. Those are all things that happen inside the system, in the terrestrial ecosystem. And so when you look at this, you have relatively few inputs. Nitrogen fixation is a huge one. That's where the bulk of our nitrogen comes into terrestrial ecosystems. Once it's here, it just moves around a lot. Animals consume plants, so we have herbivory. Plants absorb nitrate and ammonium from the soil, that's assimilation. When plants and animals die, that's death and litter fall. That dead organic matter then gets decomposed. When it gets decomposed, it's basically the same form of nitrogen that it was before. It's just that now it's been freed up from the cells that are holding that nitrogen in. So now rather than cells inside of the bodies of organisms, it's dissolved organic nitrogen, which is what we find DON is. That nitrogen that is in the form of amino acids and nitrogenous bases and things like this, then gets ammonified. That ammonification results in ammonium. That then can be assimilated by plants. It can be assimilated by microbes, and it can also be broken down into nitrate, which also gets assimilated. And then all of these things can be leached out and go into groundwater. As soon as it's in groundwater, it flows out into a stream. That stream flows into a river. That river flows into the ocean, and it's gone from the system. Then we have denitrification, which is, once again, bacteria working on nitrate to send it back to the atmosphere through sending back to the atmosphere in the form of nitrogen gas. And then we have volatilization from burning of plant communities on the surface of the earth. We also have excretion. Whenever you go pee, a big component of your urine is actually nitrogen containing compounds as well as salts and things like this. That stuff gets into the soil, becomes ammonia. That ammonia can be volatilized also back to the atmosphere. So think about the nitrogen cycle in a couple of ways. Relatively few inputs, relatively few outputs, and a lot of intra-system cycling. So things I will not do to you on the test. I will not give you a blank sheet of paper and say, draw the nitrogen cycle. That's not really what we want you to get out of this. But what I might do is I might give you a nitrogen cycle and I might leave certain sections of it out. And then you would have to put certain parts of the nitrogen cycle in there where pieces are missing. I might put a nitrogen cycle on there for reference and then ask you a series of questions about the nitrogen cycle. So I might give you the full nitrogen cycle and say, okay, well, what organisms are responsible for denitrification? It's not obvious in this chart what organisms are responsible for denitrification, but who's responsible for denitrification? Bacteria are. With a nitrogen cycle, bacteria are kind of like Jesus in Sunday school. It doesn't matter what question your Sunday school teacher asks you. If you don't know the answer, say Jesus, and chances are you're going to be right. Same thing with nitrogen cycling. A lot of this is driven by bacteria. What's a transformation that is not driven so much by bacteria? Or a flux. I guess it's not really a transformation, it's a flux that is not a transformation. Not leaching. Hmm? Fixation is accomplished by bacteria. You notice I have the fixation arrow going to the roots of plants. That's where the bacteria are in the roots of legumes.
Okay.
And then also soil bacteria occur in the soil compartment.
Volatilization. Fire volatilization.
No, that's not accomplished by an organism though. So one set of organisms is bacteria. Bacteria are involved in lots of things. Ammonification, nitrification, denitrification, that's all bacteria. Nitrogen fixation is all bacteria. What's the other group of organisms that we talked about that's important in fluxes of nitrogen? Fungi. Where are fungi involved?
It was the assimilation.
Assimilation. What types of fungi are those?
The micro, or the microhiza is like the abbreviation.
Mycorrhizal fungi.
Yeah.
Good. Good. Trevor, give your group a point, even though you got a marker.
I'll find one.
All right. So these are the kinds of questions that I might ask you about. I'm never going to ask you to produce a full-blown nutrient cycle on the board with all the numbers in it and everything else. That's just way more information than you need. But you need to have facility with how to interpret those things, and you need to be able to answer questions about the cycle while not being able to reproduce it all by yourself. Okay? Questions? Yeah.
So like with this slide, would
Uh-huh.
you expect us to be able to like put them into categories like this?
Yeah, so you should definitely know the difference between poles and fluxes, but also within those fluxes, you would need to know where those fluxes go on a diagram, and you would need to know which of those fluxes represent inputs into the system, outputs into the system versus cycling within the system. So the same kind of stuff that I just worked with.
Are those the only inputs and outputs that we're like going over? Or
I think so. Can
Okay.
you think of anything that I'm missing, Dr. Judd?
Nothing. No, we're trying not, no, I can't.
Yeah. He's thinking of more complicated things, and he's like, yeah, we're not gonna go into that level.
Yeah.
Okay, all right, yeah. This is the level that we're gonna ask you about. Alright. You have any questions? Let us know. We're gonna meet on Monday before the test on Wednesday, so we can answer those questions very well. Dr. Judd's gonna finish up. He's back there feverishly editing so that as I give him less and less time to get his job done.
You will notice that I changed the title to this if you're downloading it from Moodle. In part because we're going to focus on the prairie and nitrogen portion of this slideshow. We'll probably won't get to the carbon portion of it.
Which I've already talked some about carbon.
Yeah.
So.
Yeah, the title says, on Moodle says something like carbon and nitrogen human impacts or something. So these are the three things that I want to focus on with the time we have before we switch over to a little bit of open discussion. We want to talk about some of the ways that human activities have affected the nitrogen cycle and that our society has to deal with. So we're going from the small scale nitrogen cycle, thinking that you're doing for Kanza, and we're going to think about some practical, more societal consequences of that, especially the consequences of excess nitrogen in aquatic ecosystems at both small and large scales. And then we're also going to talk about, we're going to link this to changes in prairie ecosystems. You all have seen versions of this kind of a map several times now, right? We've got the general gradient here. What's driving this gradient from east to west? What resource?
Weather.
Weather, but what aspect of weather?
Rainfall.
Rainfall, right? And then this gives you what other aspect of weather, or climate, I should say.
Temperature.
Temperature. This is a map of the historical range of prairie, approximately, during sort of pre-settlement times from Europeans. So we've got tall grass and short grass and mixed grass prairie. We've got this thing called the Prairie Peninsula that extended almost all the way to Ohio, which is mostly tall grass prairie. And the rest of the stuff over here is mostly forest and mostly desert montane ecosystems further west. Let's look at this kind of at a more local scale. I've got a few different states to show you just briefly. This is a map of the state of Iowa where yellow represents different kinds of prairie, pre-settlement prairie. And the green represents mostly woodlands and brush, basically woody vegetation, the kind that, as we've talked about, you'll see along the water courses. Even at the small scale of Kanza, you'll see that, but even at the state level scale of Iowa, you'll see that. But this isn't what prairies look like in Iowa today. Much of that land has been transformed. Not just the prairie, but also the woodlands. And so here's a map showing you basically how land is used in Iowa today. It's mostly agriculture, and mostly the top two crops in the United States, corn and soybeans. You can also see some of the different cities for human settlement. You can still see there's woodland along the rivers. But notice how much less yellow there is, and it's little pixels. That's the region that we would call grassland or prairie today. Some of it maybe isn't undisturbed. Maybe it's been grazed heavily, or it's been plowed and returned to grassland. But notice that the yellow, the prairie environment, is much, much lower. If we look at Kansas, I didn't find a good map showing pre-settlement distributions of prairie, but this map shows you sort of where we expect those ecoregions and those communities to have been located in pre-settlement times. So of course this doesn't show sort of the effects of Native Americans so much, but which would have certainly been present. But notice all of this transition, mixed tall grass prairie, and then shorter grass prairie as you move to the west.
Notice that there are a bunch of colors on there, but the colors are short grass prairie, mixed prairie, tall grass prairie, sand prairie. So that's a lot of prairie.
Yeah, they call it a sea of prairie, right? We'll talk about that. Let's look at land use patterns today. Can you guys find the Flint Hills in Kanza? That's right in here. But when you think about the grasslands that are remaining, the green that's interspersed or speckled among the cropland, again, that's remnant prairies and also grasslands that are not being actively managed as cropland. So not all of that is going to be high quality until deep soil prairie. But notice how much less there is, right, compared to what would have been there. Just because of the total area of all this cropland, in addition to some urban usage, some grazing lands.
So,
And-
question.
Oh yeah, go ahead.
Can you remind me why the grass is like taller, that way, and then the grass is shorter on the left? even
Yeah,
though-
here's Kansas, right? There's our east-west gradient. And someone actually helped us just a few minutes ago. Someone else, what's the main gradient here from east to west? Rainfall. Generally, plant height and total plant biomass is correlated with rainfall.
Oh.
So it's just wetter over here than here, and so it supports more tall grass prairie.
Does that have to do with the mountain range?
Oh, why there's a gradient at all? Yes, because we're in the middle latitudes here. The air is mostly coming from this way, or air mass is coming up from the Gulf. Circulation is influenced by the
Yeah, just the main reason
But
for
the main
this
reason-
gradient is that you have that warm current off the east coast. Warm current, a lot of humidity. As that humidity comes into the United States, it basically drops all of its rain on the eastern half of the United States, and it dries out as it goes further and further west. So it's that you're far away from a warm ocean current.
Oh, yes, sorry, yeah. This is not simply a rain shadow that covers a third of the continent. It's also this rainfall sort of dumping it all. By the time it gets over here or up here, it's much drier. Or closer to the front range, it's much drier here. And I tried to find one for Missouri as well, and this is a map that you'll find all over. Missouri Prairie Foundation and other websites present it. This is from a 1980s study that estimated original or sort of pre-settlement prairie in Missouri. And I tried to find maps of remnant prairies for the whole state. I was out looking for remnant prairies with a student a couple years ago for an undergrad research project, and all the maps are small and zoomed in because the remnant prairie patches, especially the ones that were never plowed, are so small they hardly show up on a map. So this is a map from the Missouri Prairie Foundation. This just shows the prairies that they manage. This is not complete, but it's basically the remnant prairies are, for the most part, so small that they don't even show up on a map like this. Which should make some sense. It's a lot more wooded in most of Missouri than in Kansas. This is all part of a much broader pattern. If you think globally for a moment, think about the world's biomes: tropical forests, savannas, grasslands, boreal forests. And we look at how much of those biomes, the total land area in those biomes, has been converted into some other use, some other human use, whether it's managed forestry or cropland or urban usage. You can see that some of these broadleaf forests, subtropical forests, so tropical places with a dry season, and especially grasslands of the world, have been largely converted over to various human usage. And then you can also ask how much area in those different biomes has been protected to be somewhat natural or somewhat wild. And you can see that the highest ratio in terms of conversion to protection, the lowest ratio, is in grasslands, which includes North American prairies. So there's a lot less prairie than there once was. Most of the grasslands of the world have been converted to agriculture. There are other reasons that you might convert land, but here, most of that story is agriculture. And that's what links us to the nitrogen cycle that you just learned about. This is a diagram, another way of depicting the nitrogen cycle, that shows estimates of the inputs of synthetic fertilizers, especially nitrogen, or particularly nitrogen, and also inputs from symbiotic fixation, so bacterial fixation, soil fixation, and atmospheric deposition when volatilized nitrogen returns to the atmosphere from fire. And you can see these arrows, the width of the arrows tells you a little bit about the flux. I mean, they give you the numbers as well, but the width of the arrows gives you a visual representation. So you can see plants in grasslands and croplands are using nitrogen. It's being input into livestock and fisheries. It's going into organisms like people as animal products and vegetable products. It's exiting the soil system, leaching as runoff. Now we'll jump forward in time about 50 years.
Note that this is from 1961.
61. So Dr. Quincy told you about a process where humans do the nitrogen fixation. And it was named after a pair of scientists who developed that process. Anyone remember that from the day before yesterday? The Haber-Bosch process. That's where humans do on an industrial scale nitrogen fixation to supplement soils instead of simply relying on fixation by bacteria, by legumes. Whereas traditionally, you might have to cycle or alternate between a crop like corn and a crop like legumes. Here you can just, with synthetic fertilizers, you can just input that into the soil and grow the same crop every year for several years. This is closer to what it looks like today. Look at all this increase, how much the synthetic fertilizer input has increased. So we have much larger cropland. Much more of that gets lost as runoff. Much more of it is going into the food that we all eat, supporting the much larger population. Compared to the 1960s, what was the human population on Earth then? It's like 4 billion or something. That was about peak population growth in the 60s, and now we're approaching 8, right? So that's a lot more of our livestock, our animals, our fisheries, our people. And then there's a whole bunch of it that's, that moves around another flux here in international trade. So not all of most of the nitrogen that's produced today and used as fertilizer is both produced and used in Asia. Some of that comes over here and we use it on our farms as well. So what are the effects of that much larger runoff arrow that we had at the bottom? That's the question we want to talk about here. What's the effects of more anthropogenic nitrogen inputs? Fixed nitrogen created for fertilizer and nitrogen oxides produced by combustion, that volatilization, gets dramatically increases the availability of nitrogen in ecosystems for photosynthetic organisms, not just plants, but what's a photosynthetic organism in aquatic environments? What are photosynthetic organisms in aquatic environments?
Algae.
Algae. Which is a big and diverse group and fascinating group, but we'll just say algae for now. Paul has it up on the board now. We've more than doubled since 1961. 1961 is right about when human population growth was fastest it's ever been. So as nitrogen availability goes up, the productivity of photosynthetic organisms tends to go up. Why? What are they using nitrogen for? For photosynthesis, what part? What kinds of things have nitrogen in them in the bodies of plants and animals and algae? Where do you find nitrogen? Link back to those macromolecules you were talking about. Not just the leaves. Think back to the macromolecules that we listed on the board.
That's what the roots.
Oh, sorry.
The roots.
Roots still plant organs. They're going down to macromolecules. We talked about lipids. What else did we talk about? Carbohydrates. Where's the nitrogen? In the proteins. Proteins and DNA. And what are some important proteins involved in the carbon cycle and the nitrogen cycle?
ATP.
No, ATP is not a protein. Catalyst, but in a biological sense. Enzymes, right? What's the important enzyme in the nitrogen cycle? The fixing bacteria use? Nitrogenase. And what's the important enzymes involved in carbon capture in the carbon cycle, or photosynthesis? Rubisco is one, and what's the other one? Carboxylase. Good. So we need nitrogen for these things, right? So as nitrogen availability goes up, you can make more of these enzymes. So they're going to capture more carbon. So they need it for the enzymes and for carbon capture.
Dr. Judd?
Yeah.
Do you carry around in your head that Rudisco is probably the most abundant enzyme on the planet?
I do. I remember making a, I think I said that on a photosynthesis day. But yeah, it's partly because, and we'll talk about this, I'm expecting a review question on photorespiration, so that might come up again before the end of class. Ultimately, though, you can get to a situation where there's unused nitrogen. Adding all these inputs, you can get what we call nitrogen saturation. This is a map showing the distribution of excess nitrogen deposition in the United States. And you can see that a lot of it is in kind of our breadbasket, where we're doing a lot of fertilization of cornfields, soybean fields. Mostly, we're talking about things like nitrates and ammonium. The consequence of this is fertilization. So total nitrogen in, it can be measured or can be kind of approximated by the yield of things like algae and plants. And notice that as where nitrogen deposition is highest, the productivity of plants is also highest. And most of that is coming from corn and soybean crops. So that's excess nitrogen that's leaching off of all this agricultural fields that once was prairie. Yeah.
Is that why farmers alternate crops?
Yeah. So one of the benefits of alternating crops is that you can apply a little bit less fertilizer to the soil and have less leaching because you're letting bacteria do the input of nitrogen instead of buying a bunch and having to do the math on how much should I put per acre and how much is that going to affect my crops and how much is going to leach out or waste my money. Yeah.
I was listening to the news yesterday, and have you guys heard about the supply crunch that's happening that's driving up inflation? Supply chains are being messed up because of the pandemic. Evidently, fertilizer costs are 2 to 3 times what they were a year ago, and a bunch of farmers
Because it all comes
in
from
the
Asia.
Midwest. What's that?
It's because it all comes from Asia now.
And a
Part
bunch of
it.
farmers in the Midwest are going to not be planting corn this year because they're going to be planting soybeans instead. Because if they plant soybeans, they don't have to fertilize the crop as much because once again, soybeans are making their own source of nitrogen. And so expect a shift in agriculture next year due to the expensive fertilizers. They're going to be shifting to kind of self-fertilizing species of plants.
That'll echo across the economy for a few years.
And that will echo across the economy because we use corn to make ethanol, and it's going to be a mess.
Yeah.
No. Can't happen.
So this brings me to something that I want you all to have a sense of, which is the effect of this kind of land use change on the world's grasslands on the world's aquatic systems. So I want to distinguish between an oligotrophic system and a eutrophic system, or an oligotrophic aquatic ecosystem and a eutrophic one. And the difference here is that the inputs of biologically available nitrogen and phosphorus, and also sediment in this case, tend to be low in an oligotrophic system. So the inputs of these important nutrients like nitrogen and phosphorus tends to be low in an oligotrophic system, whereas nutrients are highly available and highly abundant, and the rate of input is high, the flux in is high in a eutrophic system. In a eutrophic system, we have some consequences here. So how many of you have ever seen a pond or a lake or a waterway that looks like this? So fertilization from runoff, from leaching or just runoff, especially from farms, but also from people just buying a big bag of fertilizer and putting it on their lawn much more than they need, and then it runs into the sewer and down the stormwater basin that way. So big scale and small scale inputs. Increases productivity of the algae in the system. The algae then bloom or multiply, covering the surface of the water. Highly productive, high yield in terms of biomass, blocking some of that light, and also that biomass eventually starts to die and sink to the bottom and starts to decay. What kinds of organisms are involved in the breakdown of decaying plant biomass? Bacteria, right? Perfect, right? What are all those bacteria using as they're metabolizing and breaking down this plant biomass? What do you need to run metabolism as a heterotrophic organism?
Glucose.
You need glucose, for sure, but that glucose is actually coming from the breakdown of the plant matter. Say again? Again, that's stored in the biomass they're breaking down. These are not wrong answers, but what else do they need?
ATP.
They do need ATP, but they're making that from the glucose, right? The energy that's in ATP is coming from the breakdown of the glucose.
Cellular respiration, what's not? Not, this isn't photosynthesis. What?
Oh my.
Yes.
Wow.
So all these organisms that are in the water are using oxygen that either dissolves normally from the atmosphere or is produced by the plants that are in there. But the rate at which oxygen diffuses into the water and the rate at which these plants produce dissolved oxygen in the water is pretty slow. You've got tons of plant biomass and other dead and dying things at the bottom of this pond or this waterway. And all that dissolved oxygen is being used up by the bacteria. The water becomes anoxic. And all the fish die, all the, most of the macroalgae die because they don't get light. Aquatic invertebrates don't have enough oxygen. So the system collapses and it becomes a very ecologically simple system of algae, decaying, and bacteria. And that's all you get. So you don't have all the fish, you don't have all the turtles, the snails, all these organisms that you think of when you think of a healthy oligotrophic water ecosystem or aquatic ecosystem. And it's mostly driven by the loss of dissolved oxygen in the water column due to cellular respiration. So you all have seen that on the small scale. It's also happening at much larger scales. The Mississippi River watershed, if you've thought about watersheds at Kanza where they burn one or another, of course those are little pieces of a much larger watershed that drains one of the largest watersheds on Earth. I don't think it's the largest, but probably top 5. The Mississippi River watershed. And so the area around the Gulf of Mexico, around the coast of Louisiana where the Mississippi River drains, fluctuates and sometimes becomes anoxic for long periods of time. In certain places, stay anoxic for a long time. So the redder it is here, the lower the concentration of dissolved oxygen in the water. And so these are called dead zones. And they happen around estuaries around the world. And you can see all this gray stuff here in this estuary. There's trees in the background off the coast of Louisiana. That's all fish that died because they didn't have enough oxygen.
Wow. I thought that was like rocks.
It kind of looks like it, doesn't it? This picture, I took this from your textbook, so you can see, you can reference the caption for this.
An easy way to fish, though.
Don't think you, I don't think you want those fish.
That's like driving down the highway and seeing a deer, like, oh, it's still good, it's just bruised.
Ew.
You don't want to eat that, that's gonna be bad.
You don't eat first meat.
You know? So it happens not just here at the mouth of the Mississippi, but at many of the world's major estuaries and deltas, especially those that drain the largest agricultural systems. Agricultural systems across Europe, across North America, in Asia, especially in Asia. All right. So the rest of the slides that I have here are all about carbon cycling. So I encourage you to take a look at this stuff just to think about it. I put in some slides here, but please scroll forward. Well, before I end here, so questions about eutrophication at the small scale or the large scale. We haven't talked about the oxygen cycle. It's mostly atmospheric, but the availability of oxygen becomes relevant when you think about species interactions and nitrogen fertilization. Yeah.
So can you like define eutrophication?
Yeah, let's see. Well, eutrophication is the consequence of nutrient loading. Let's see here. Did I have a definition for it here? I guess I didn't. All right. It's nutrient rich. It's okay. Nutrient fertilization of aquatic ecosystems that leads to anoxia. That's what we want for definition. I can look one up too. The consequence of this.
You can erase all the rest of that. Gary used eutrophication to describe terrestrial systems in theory, but I never heard anybody use it in reference to terrestrial systems. It's always used in aquatic systems. Probably because terrestrial systems don't ever produce oxygen to the point that it's a problem.
So the link here between anoxia and the collapse of the food web, the loss of all those animals for live dissolved oxygen, and the nutrient fertilization is what I want you to understand. And that's what's in the text on the slide. So make sure you understand what those bullet points are saying. Okay, well, I'm just gonna have to scroll through, so. Okay, yeah. All right, if there's no more questions on eutrophication, we're at 1:16. So let's open the time now for some review questions, or if you want to talk about putting the finishing touches on your DNL summary, this is your time. Let's make the, yeah.
So for the prairie forb animal and grass of the day, how are those going to be presented on the test?
Oh, often we'll use those prairie organisms of the day as kind of characters in some of the questions. So you want to know some of their important functional attributes. So know about the C3 and C4 organisms, know about organisms that played a role in nutrient cycling in some way that we talked about. We're not going to test you on the 18 that we've covered over the course of each lecture. But they make appearances in questions too, generally. So look for their functional roles in prairie ecosystems. Yeah.
So I just clicked ahead on your slideshow. What does the different letters above bars indicate significant differences?
Yeah. Well, so I wanted to start by asking you a question. What do these two bars show? Just look at it. What are those two bars showing? Yeah, start us off.
The darker one was burned annually, and then the lighter one was burned once every 4 years.
All right, and what is this graph? So that's correct for the colors. What's this a graph of?
Species richness, so the number of species in the area.
And which, I only circled two bars with a red square, or I only indicated two bars with a red square, so which two did I indicate?
Ungrazed plot.
Yeah, ungrazed. Okay, so someone other than Sonia, are those two bars the same height?
No.
Somebody read, now you said no, somebody read this out loud for me, someone up front. Kate, can you read that?
The different letters above the bars indicate the significant differences at P is greater than or equal to, or less than equal to 0.05.
Does the difference in height between these two bars matter?
No.
So that difference is natural variation within a system that where species richness doesn't significantly vary from 4-year burns to 1-year burns.
And why do they have to give you the letters to indicate to you what's different? Because they're plotting standard errors of the mean rather than what?
The exact.
Say again?
The exact, like, without error.
Well,
the
there's only
actual
there.
value is the height of the bar.
Yeah, the height of the bar is the average.
What other kind of measure of variation could they have presented?
That is more informative.
95%.
Yep. This is why I'm an advocate of 95% confidence intervals. If you have 95% confidence intervals, you might still add the letters to indicate to somebody, but you don't actually need them at that point. Because a 95% confidence interval would be wider, and as a result, encompass the other mean. Especially with only a sample size of 4, which is 3 degrees of freedom.
Yeah, it's going to be
which is like gonna be pretty big confidence interval.
So what's going on with BC, C over there on the bottom right?
Over in this panel,
Yeah.
yeah, well, is the dominance index, so since it's D, different for ungrazed that were burned every 4 years and grazed burned every year? Are these two bars significantly different from each other? Actually, let me step back here. So these have the same letter, right? So they're not significantly different. What's a little bit easier to do first is to look at maybe one of these. So let's look at the diversity. So what was the diversity index that we used when we talked about in class? Yeah, Shannon's H, right? So is the diversity different in grazed plots burned every year, every 4 years?
No.
No. If you're in an ungrazed plot, does the burning interval, the fire regime matter for species of richness?
Yes.
So let me ask you the same question again. Does fire regime, burning regime, fire frequency matter in a grazed plot for diversity?
Not really, no.
That's an interaction effect. They talk in the introduction about how people weren't investigating interactions between these two things. That's what they're looking for here. They're looking for interaction effects. So we're also seeing an interaction effect down here. Do grazed plots have similar or different dominance scores? Similar. They both have a C. Do ungrazed plots have similar or different dominance scores based on fire frequency?
Different.
That's an interaction effect again.
One of the ways that it's easy to detect whether an interaction is occurring is if when you draw lines between, in this case, ungrazed 4-year burns to grazed 4-year burns versus ungrazed 1-year burn to grazed 1-year burn. If those lines are parallel with one another, it means that the grazed plots are behaving the same way the ungrazed plots are, and fire is also acting the same way in both of those. If those lines diverge from one another, or converge on one another, or cross one another, it means that something is happening in grazed plots that's not happening in ungrazed plots in terms of the interaction with fire. And so I generally find these kinds of plots to be more productive in thinking about interactions, because here, if you had had the plot that looked like this, in this case, grazed plots have higher dominance than ungrazed plots. If dominance, or it wasn't dominance, it was diversity, was it? Diversity was the one that you guys were looking at. If you have higher diversity in the grazed plots, you have lower diversity in the ungrazed plots, but the relationship between 1-year and 4-year burns is the same. So fire doesn't interact with grazing at all, because what's happening between the fire treatments is the same in both grazed and ungrazed. The difference really is just that grazed has a higher diversity than ungrazed. So one of the things that I've done, I have actually a copy of this paper where I've actually drawn all the lines between the peaks to see if they're parallel to one another, if they're crossing or diverging. But that's another way of thinking about interactions. I don't know what happened.
I was just trying to scroll to one of the other fingers on Beam where we could try that, but now I've lost communication with the.
That's weird. Oh, you got your play back. There you go. Yay.
So those are two lines with similar slopes, do you think?
Yeah.
So when we're writing about looking at altitude, would you like us to focus mostly on heterogeny, or do you want us to focus equally on heterogeny and the resource variability? Like, how much do we need to talk about resource variability?
Well, let's unpack heterogeneity. I think that's going to be key here. the heterogeneity, you can think of it as spatial variation, but there's spatial variation in lots of elements, lots of aspects of there. So, were any of the graphs in the results, did any of them have heterogeneity as their y-axis?
No.
Dissimilarity. Let's think, so, there were three elements of diversity or community structure that we talked about when measuring diversity data. You guys remember what are the components of community structure or diversity? Give me one of them.
Species richness.
Species richness is one of them. Can someone else define species richness?
How many species are in a given community area?
Yeah, you count them up. How many species are there? What's another aspect or dimension of community structure?
Dominance.
Dominance is correct, but let's generalize it. Evenness is kind of, think of evenness as the converse of dominance.
Relative abundance.
Abundance. Abundance is the other element. Do you remember what this kind of graph was called? What do we call this graph? Rank the species.
Species. Rank abundance.
Distribution.
Distribution.
Is there a question on the test review packet that graph?
There is one of these on the old exam that we posted on Google. What's the other component of communities? It's not captured here by number of species and how abundant they are. Well, these two things combined is what gives us Shannon's H, which is the diversity index. But there's something that Shannon's H doesn't capture.
Species composition.
Composition. Who they are. Which figure in the VNL paper compares composition? Let's look up here and then see if the answer becomes obvious. That was a good graph.
You want being back?
No, I thought your graphic might make
Oh.
it easy for them to figure it out.
Oh, yeah, that's why I pulled it up. Oh, where did it go? Ah, there it
Oh,
is.
you're on my, nice.
Yeah, I'm on your computer. I'm doing stuff on your computer.
That's all good.
So here we have each of these colored dots is a different species. This is one of my favorite questions that I've ever made on a test. Each colored dot represents a different species. And so we have 8 species here, 8 species here, 8 species, 8, 8, 8, 8, 8, 6, 7, 7, 7, 7, 7. There are either 6, 7, or 8 species here, so species richness is lower here than it is here, because here we have 9, 9, 8, 8, 9, 9, 8, 9, 9, 9, 10, 10. But when we think about heterogeneity, what do you notice about all of the plots in this sampling unit versus all of the plots in this sampling unit relative to the colored dots that are there, the species that are there?
The one in the enclosure is the same colors.
The one in the enclosure, they're all the same, except for the ones that are additional out here. They're all the same. That one, 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 that one. Same, 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 same. The only thing that differs is what's off to the right. What's happening over here?
They're very
Every one of them is different from every other one. Which of these areas, the one in the enclosure or the one in there, which one has the higher heterogeneity? Yeah, the left one. That's what heterogeneity is about.
There's a slight difference in richness here. There's minimal, if any, difference in the rank abundance, because we're only dealing with 8 circles, roughly. But there's a big difference in composition.
Composition from plot to plot to plot to plot to plot to plot. That's what heterogeneity is about. This plot looks different from that plot looks different from the previous plot and the next plot.
So now.
These will look very similar with little small tweaks.
So they don't have a composition axis on any of the graphs, but which graph captures or includes variation of composition? Yeah. What's that caption say?
Where did your thing go? Where is the other? Oh, there it is.
On the first page. Oh, it's right there.
Oh, there it is. Yeah. I scrolled through it.
So look back at where they define heterogeneity and community composition. But notice that the word is there in the caption, whereas dissimilarity is what's on the axis. And you'll see how they calculate dissimilarity as a measure of heterogeneity in the text.
Yeah, so remember when you're reading a figure, there's always the figure number and then there's a title. And you may or may not latch onto what the title is saying immediately, but because it's talking about heterogeneity, that tells you what it's about. But really, they're measuring heterogeneity in terms of how similar or dissimilar one plot is to another. And so then they explain that in the explanation down here.
Similarly, they could have put H as their y-axis. They could have written Shannon's H instead of diversity, because that's the metric they're using. Other questions?
So you don't want us to talk about resource variability?
Oh, sure.
Oh,
We can
sure,
talk about resource
yeah.
variability.
I just wanted you to understand that heterogeneity refers to variation in species richness, abundance, composition, and resource availability. All of those things contribute to spatial heterogeneity in the prairie. So unpack this idea of heterogeneity and talk about their results. And they give you results for each component of spatial heterogeneity, each resource, dissimilarity, diversity, abundance.
This is how much nitrate there is. This is how heterogeneous that nitrate is distributed. This is how much light there is. This is how heterogeneous that light is in the prairie.
Oh.
Because it's the coefficient of variation. It's how much variation there is from spot to spot in the
And
prairie.
they define that V statistic in the text.
And also, we went through that at the very beginning of this case study in the other book.
Okay, so the heterogeneity is like an umbrella, and then under the umbrella are the coders like.
They have heterogeneity of all kinds of things, yeah. And they take you through heterogeneity for species, and they take you through heterogeneity for nutrients, or resources rather, one nutrient and then light as a resource. What's that?
I said solid.
Okay.
We're about out of time, but I noticed that I was kind of giving a lot of like 7 or 8s or 9s out of 10 on the photosynthesis homework. So I'm gonna make a post for you guys tonight. Hopefully I'll have that by tomorrow. Just on photosynthesis. But if you want to supplement the videos that are on YouTube, look at section 16.4, I think, in your book. So it's in chapter 16. Skip forward to the section where you'll see the bundle sheath cell diagram. And review that. Get yourself another way of engaging with the material, not just listening to me talk, but also looking at that section on C4 and C3 in the textbook.
Exam 3 from last year is up on the Moodle site. That's where that question with the colored dots came from. After talking to Trevor the other day, I went looking for old exam 3s, and I went back to 2015, 2017, etc. And I think the one from last year probably reflects better how we're teaching now than something from 2015, so I put that one up there. Even though it spoils what has been one of my favorite questions that I've ever read for a test. So, anyway, it's on there so you can use that as practice. Also, at the end of class on Monday, after you discuss being as a group, I would be shocked if we don't have some time to also